YouTube live stream.
Oh, that's
This is
smart.
a YouTube live stream.
My family's watching that live from literally on a boat in the middle of the ocean.
Really?
Yes, they're shrimping right now.
Oh.
So it is live now.
Huh? They have service?
Yeah, I'm amazed that they have
Yeah.
Sarah,
Wow.
it is
It's
live,
live
now.
now.
So just check whether they can.
Hi everybody. No one's probably out here yet.
Dark.
Yeah.
You don't have a clicker on you, do you? I can use my computer.
They want to turn some lights on it. That's
Yeah.
kind of dark.
Okay. Yeah, I'm just a little. I have like staining pictures. Is that better or worse?
No, it's dark as it is. I would probably leave that open. It's dark on that screen.
Oh, gotcha.
Could you like text your family or and here?
Yeah, it's like, you know, you're hiking. How much you just know? They're running out of energy.
Oh, I'm like, you're not, you're definitely not your first. Who messed up? Should I get those? I was really cool.
Alex Baking Services. I'm still trying to.
So they have a bit of a pad thai noodles like around that. I think there's yeah, and that's what we're and he was like, and he like, this other girl wanted, she was like, okay, which class we're going to? She's like, what have you been up to recently?
I'm like finishing up basically my apartment every time I see you, I always get reminded of the key. It's like
like I just got and like, all right.
maybe I'm
Yeah.
probably.
Okay, so we'll go ahead and get started. Everybody's here. So it's a real pleasure to introduce Sara to give her seminar in defense today. Sara and I met about 5 years ago. I'm going to tell one little story. And it's not from a conference, I promise. We're going to tell. Sara and I initially met about 5 years ago in 2014 in Vancouver. At the time, I was on the East Coast, and she'd approached me about working with me in South Carolina. And so we ended up meeting in Vancouver at the CTEC meeting that was up there. And then right about the time, around the same time as when I was looking at UCR to apply and potentially interview. Anyways, a few months later, we were talking on the phone. I don't know if you remember this, but talking on the phone, and I hadn't heard yet about, I think I got a verbal offer or something. It was around March. And we were talking on the phone, and I had asked her, if she had applied to any other places, and I hadn't mentioned anything about potentially coming out here. And she's like, listed off a few things and said UC Riverside, and I was like, oh, that's interesting. So I investigated a little bit and was trying to determine whether or not to tell her that I was potentially going to come out here. Anyway, so I ended up telling her that, and so she ended up coming to UCR. I ended up coming here, and she took a very risky move to work with me, mainly because starting up a lab again is a risky sort of enterprise, and a lot of things can go wrong. And Sara, really out of the group right now, she's the only one that kind of saw the lab, from being an empty lab with a bunch of rocks in it that we had to get removed to what it is now. And so Sara has a unique perspective in terms of kind of what it takes to run a lab, and as a result, she played a big leadership role in terms of training other students with fish husbandry and a lot of other things. And so it's been very much appreciated. Sara's been an incredible asset for the lab and the department at UCR. Trying to think what else. Oh, the other thing was, so I presented her with a couple different projects in the beginning, and one of which was sort of this area of high throughput screening. And I said, well, you can, choose whatever you want. And she came back and she said, yeah, I'm really interested in the high throughput screening stuff. And I said, well, I don't really have funding for that right now, but sure, go for it. And she went for that. I tried to get some funding a few different times. It didn't pan out for the particular project she was working on. But luckily, she got on T32 support for a couple of years, so that covered her. And then I was able to piggyback off of my NIH grant and some other funds to keep her project going. And so as a result, as you'll see, her dissertation is one of the few that's actually cohesive and kind of leads from one chapter to another. Typically, it's sort of piecemeal and you're kind of, mashing things together. And a lot of that is driven by, the needs, for grants and things like that. So even though it was a struggle over the last 4 years to kind of, make sure we had the money to keep it going and there was some uncertainties and so on, I think it ended up being a really nice piece of work. So that's all yours.
Can everybody hear me?
Yeah.
Okay, awesome. All right, thank you so much for that great introduction, Dave, and thank all of you for being here today. I know 9 a.m. is early for some of you, and I really appreciate it. I'm excited to talk to you about some of the work I've done doing my 4 years at UCR. Okay. So today I'm going to tell you a story about how we've been able to leverage zebrafish to identify chemicals disrupting early embryonic development. So a quick outline, I'm going to start with a brief introduction and overview of my research aims. I'll then go into each of the 3 objectives I'm going to talk about, and then end the presentation today with some overall conclusions that we've been able to draw from this work. So jumping right into the introduction, globally there are thousands of chemicals produced every day. In 2018, global chemical sales were valued at over $4 billion. This infographic you're seeing on the screen here is showing you the distribution of those chemical sales. And as you can see, as of this 2018 report, the majority of those are happening in China. And this chemical production is expected to continue to grow by around 3% each year. So this growth in chemical production and the redistribution of chemical production around the world raises a lot of questions about the testing of these chemicals for their safety and chemical regulation. So in the United States, chemicals are regulated depending on their use. Pesticides, for example, are regulated under the Federal Insecticide, Fungicide, and Rodenticide Act, more commonly called FIFRA. Because pesticides are designed specifically to be bioactive, they require hundreds of studies to assess their safety before they're allowed to enter the market. And these studies include mostly mammalian studies, such as rat, mice, rabbit studies. And these chemicals are required to be reevaluated every 15 years to account for advances in the sciences. Industrial chemicals, on the other hand, like things like paints and plasticizers, are regulated under the Toxic Substance Control Act, or TSCA is how I'm going to refer to it. TSCA was first enacted in 1976. And when this came into law, there was already around 62,000 chemicals already being used in commerce. And when TSCA was enacted, these existing chemicals were allowed to remain on the market and are generally presumed to be safe until there's data suggesting otherwise. New chemicals that have come onto the market after TSCA was enacted are required to be tested for safety by the manufacturer. However, there's minimal specific toxicity testing requirements, especially relative to the different regulations. So due to concerns surrounding the lack of safety data for many of these industrial chemicals, the Lautenberg Chemical Safety Act was passed in 2016. You may have heard of this referred to as TSCA reform. What this act does is it allows EPA to require the testing of these chemicals and generate risk-based safety standards. So it's great that we are trying to reevaluate these chemicals for their safety, but when you think about what's actually required to test around 85,000 chemicals, it's a pretty daunting task, especially when you think about these traditional mammalian models. It raises a lot of animal use concerns. And for that reason, there is increased interest in the development of alternative toxicity testing strategies. At the bottom of the screen here is a quote I pulled out from the TSCA reform guidelines, just highlighting the kind of the push to go away from these traditional mammalian models. So as just mentioned, traditional toxicity testing using mammalian animal models are just not adequate to address the sheer number of compounds lacking safety data. Therefore, we need alternative models to help us prioritize chemicals for further testing. Really with this idea of how do we address more chemicals with larger sample class sizes using less resources. And so there's a number of different alternative toxicity testing methods out there, and they range kind of along the spectrum from high throughput, where we can work through hundreds to thousands of chemicals, all the way down to low throughput methods where we're addressing a very limited number of compounds. So on the high throughput end of the spectrum, you have computational toxicity testing strategies, as well as in vitro cell-free and cell-based assays. Representing kind of an intermediate step here are alternative non-mammalian models, such as the fish embryo models or C. elegans, stuff like that. And then at the bottom, you have those conventional toxicity tests using mammalian models. So one of the alternative models, and the one I'm going to focus on today, is the zebrafish. So the zebrafish is a promising alternative vertebrate model, particularly for developmental toxicology testing. The zebrafish has a suite of different characteristics that make it a really great model for this type of work. They have a sequenced genome, they have about 70% homology with humans. They also have a rapid developmental timeline and undergo transparent ex vivo development, as you can see by this series of time course images here. So because they develop rapidly, we can test chemicals fairly rapidly. Because they're transparent and they're developing ex vivo, we can observe these developmental processes as they're occurring and look for any alterations to developmental processes. And this is a lot easier compared to a model such as a rodent, where those developmental time points are happening in utero. It's a lot harder to visualize that. They also have a high fecundity and a small size. So if things are working properly, we can get thousands of embryos every day, allowing us to quickly run toxicity tests, and their small size makes them amenable to screening applications and various microplate applications. So now that I've convinced you how cool zebrafish are, I'm going to jump right into my research aims, and I'm going to spend some time going into each of these individually. So for my first objective, we wanted to characterize embryonic zebrafish behavior for the identification of developmental neurotoxicity. So the developing embryo, in particular, the developing nervous system, is a very sensitive target for chemical exposures. And as we've already discussed, there's minimal developmental neurotoxicity data available for a lot of the chemicals currently used today. Current D&T testing guidelines require hundreds to thousands of rats per chemical. You can use around 20 liters per chemical. You can imagine the animal concerns that quickly amount when you're thinking of addressing a large number of compounds. The zebrafish is a promising alternative model for D&T testing because its neurodevelopment is very well characterized. The zebrafish has been used for many years to study neurodevelopmental processes, so there's a good foundation to look at potential toxicity. The development of the zebrafish nervous system goes through two successive waves. It starts with primary neurogenesis that happens around 10 hours post-fertilization, represented as top image here. And primary neurogenesis is characterized by the development of these big neurons that extend their axons out and innervate axial muscles. And that happens around 18 hours post-fertilization, and this process is complete by 24 hours. So as these neurons are extending out their axons and innervating those axial muscles, you get a stereotyped behavior that begins to emerge. You can see it here in this video. It looks a little bit like a tail flip. These spontaneous contractions are the first sign of zebrafish locomotion, and they peak at around 25 hours post-fertilization when they're over 28 degrees Celsius. So these spontaneous contractions are actually not something that's unique to zebrafish. They are conserved across all vertebrate development, including humans. They are spontaneous and that they're not influenced by cues such as normal light and touch. And although their biological basis is really not completely understood yet, they have been shown to be driven in part by chemically mediated neurotransmission as well as electrical coupling through gap junctions. And these spontaneous contractions are actually used worldwide as a readout for the identification of neuroactive compounds. It's something that's being pretty widely used already. However, we run into this big question when we think about assays that are using this behavior as a readout. And that is, what molecular events can we actually detect by using alterations, these spontaneous tail contractions, as a readout? At the end of the day, we want any screening assay to be predictive of a given interaction. And so this is the question that we wanted to address with the rest of this project. So we have developed a high content behavioral screening assay using spontaneous contractions as a behavioral readout. I'm not going to go into too much detail on this right now in the interest of time, but I would be happy to answer questions about it. Essentially how this works is we have a 384 well microplate. We can array 1 embryo per well, ending up with 16 embryos per treatment solution, 24 treatments. The embryos are statically exposed in that treatment solution until 25 hours post-fertilization, where we can take this plate into automated image acquisition and assess embryo survival. We assess embryo total body area as a measure of growth, and then we can quantify alterations to that spontaneous contraction behavior. So to address the question of what are alterations to spontaneous activity actually telling us, we chose to screen the LOPAC 1280 library of pharmacologically active compounds. So the LOPAC 1280 is a library of 1,280 well-characterized small molecules. It's commercially available, and it targets a diverse set of different biological receptors and different modes of action. In addition, over half of the library targets neuroactive targets. So therefore, this library provided a really great tool for us to use to probe this behavior and see what sorts of biological targets and modes of action would give us a behavioral response. So we took a tiered screening approach to screening this library. We first screened all 1,280 compounds at a limit concentration of 10 micromolar. Embryos were exposed from 5 to 25 hours post-fertilization, and then we assessed embryo survival, body area, and alterations to behavior. And following this first year, we ended up with 102 compounds that were positive. 46 of those were altering behavior in the absence of effects to growth or survival. We then wanted to narrow this down a little bit further. So we took these hits into a second tier where we shortened that exposure duration to 23 to 25 hours post-fertilization. And following that second tier, we ended up with 15 hits impacting spontaneous contractions in the absence of other effects. So looking at these compound hits a little bit closer, going into this project, our original hypothesis was that the compounds impacting this behavior are likely going to fall within this neurotransmission targeting half of the library. So that's what you're seeing here represented by these pie charts. We have the neurotransmission targeting half and we have the non-neurotransmission targeting half. And as you can see with this tier 1 data here, there was actually about an even split of compounds that were impacting this behavior across the neurotransmission targeting half and the non-neurotransmission targeting half. Additionally, you can see by these lists below the pie charts that the compound hits were associated with a wide different range of biological targets. So we weren't really getting a huge degree of specificity here. When we took things into a second tier, we did have a larger proportion of those compounds within the neurotransmission targeting half of the library, but there was still falling within the non-neurotransmission targeting half, and we still maintained a pretty good degree of variability as far as our biological targets here. And so this really suggested to us that maybe spontaneous tail contractions are not specific. They were not associated with any particular biological target or mode of action. And this really suggested to us that maybe this behavior is affected by some downstream effects or other mechanisms of action. So then we got to thinking, what else might be influencing our results here? And one thing that you need to think about whenever you're doing these aqueous exposures with zebrafish embryos is compound uptake. And so we wanted to see if the compounds that were bioactive within our screen were distributing to a particular chemical space. So to answer this question, we plotted all of these chemicals by their hydrophobicity, which is the log P value, synonymous with log KW, by their molecular weight. And so when we look at the tier 1 data, you can see that the compounds are not really falling within a particular area of the chemical space, but when we shorten that exposure duration in tier 2, we get a narrowing of that log P range. And this was really interesting because it indicated that maybe we're biasing towards compounds that fall within this optimal log P range around 2 to 5. So the next way we wanted to look at this data was in terms of its structural similarity. So we were interested if a particular structural feature was influencing our results. So what we did here was we used a PubChem structural clustering tool, which structures compounds based on a two-dimensional structure. And I know this is really hard to see, but I just want to draw your attention to the heat map here. So we plotted all the compounds based on their endpoint. We also did this for the whole library. I don't have that data on here because it was a giant graph. But essentially, we did not see any sort of grouping with these compounds, and it didn't appear that this two-dimensional structural similarity was influencing our results. I think it is important to mention, though, that the LOPAC 1280 is inherently a diverse library. That's what it's supposed to be. And so it is possible that these results are due to that inherent chemical diversity, and we might be finding something that way. So that brings me to some conclusions for this first objective. So we showed that spontaneous tail contractions are influenced by a wide range of pharmacologically diverse compounds, and that the use of this behavior as a readout, at least within our particular assay, was unable to predict biological targets and discriminate opposing modes of action. And this really raises some questions about the use of this behavior as a predictive endpoint in all of these assays around the world, and really, I think, highlights the fact that we need to do some validation with this across the field. And I do want to end just talking about some uncertainties. As with anything, there was a ton of uncertainties associated with this assay, and this included underlying zebrafish biology. So the targets that were provided for all of these compounds were primarily derived from human cell lines. And so whether or not those targets are being conserved across the zebrafish embryo is a definite uncertainty there. Assay design could have also influenced our results, and also this issue of toxicokinetics. We didn't do any internal concentrations, so it's really hard to say if our negative results were due to a lack of bioactivity or a lack of uptake. So those are things we need to think about in interpreting this data. Right. So moving on to our second objective, which was to characterize the developmental toxicity of niclosamide within zebrafish embryos. Okay, so there was a number of hits that came out of our LOPAC 1280 screen that were really interesting, and one of these was niclosamide. So within our tier 1 screen, niclosamide resulted in 100% embryo mortality. When we took that down to the shorter exposure duration, we had 100% behavioral alterations as hypoactivity, so it just stopped all behavior. And when we went to look a little bit more into this compound in the literature, we actually realized that it's a really public health relevant compound. Niclosamide is widely used for the treatment of tapeworm and intestinal parasites within humans, within livestock, as well as within veterinary applications. It's actually listed on the World Health Organization's list of essential medicines. In addition to this traditional use, niclosamide is also under investigation for a wide range of alternative human health applications. So due to our interesting developmental toxicity results in our screen, as well as the importance of this compound right now within public health and medical field, we chose it as a compound to follow up on. So the traditional mode of action for this compound within invertebrates is thought to be the uncoupling of mitochondrial oxidative phosphorylation, or OXPHOS, and associated the inhibition of ATP production. And this mechanism really came out of work in invertebrates and in tapeworms, where this was thought to be the mechanism. Now, as I mentioned, niclosamide is under a lot of investigation right now for a number of alternative uses. And just to demonstrate this, I did a quick literature search looking up niclosamide with some of these search terms. As you can see, there's a lot of talk about it as a potential anti-cancer drug to treat bacterial infections, immune disorders, neuropathic pain, endometriosis. And I think one of the more interesting ones is Zika virus. Actually, when I did this literature search for my calls a couple of years ago, this number was 3. So it's tripled just in the past couple of years. So there is a lot of conversation going on right now in the Zika community. So within all these alternative uses, the mechanism for how niclosamide is working is really not well understood. Niclosamide has been shown to interact with a wide range of different signaling pathways and result in a wide range of different effects across different model systems. There's really no consensus for how this chemical is working. There is consensus, however, that for these alternative uses, new exposure routes are necessary. So one of the things that makes niclosamide an awesome antiparasitic drug is that it has a very low oral bioavailability. So you're able to take it orally, it's able to pass through the GI tract, preferentially targeting parasites, while not being absorbed to a large degree across the GI tract, resulting in really low host toxicity, which is fantastic. But that does pose a problem when we have alternative uses where we need to get this compound into the body. Therefore, we do need new routes of exposure, necessitating new toxicity testing profiles. Developmental toxicity, in particular, needs to be evaluated. As of right now, the developmental toxicity for this compound is not well studied. And so there is a clear need for some developmental toxicity data here. So as I mentioned with our screen, we saw 100% embryo mortality with the 10 micromolar dose. So the first thing we wanted to do here was bring that concentration down and see what types of phenotypes we were seeing at non-toxic concentrations. And so that was the first thing that we did here. And interestingly, what we saw when we got to lower concentrations is a concentration-dependent delay in early embryonic development, particularly in the process called epiboly. Now, for those of you not super familiar with zebrafish development, zebrafish develop as a mass of cells proliferating on top of a large yolk sac, which you can see right here. At about 4 hours post-fertilization, those cells in that cell mass begin to thin and migrate around the yolk sac until that yolk sac is eventually completely engulfed. And that thinning and migration is a process called epiboly. So this is what we were seeing effects on with niclosamide exposure. I mean, you can see that demonstrated here in the series of images. So now that we had a handle on the phenotype we were seeing, we wanted to dig into this a little bit further and look at the ability of these embryos to recover from niclosamide exposure and the existence of a potential sensitive window for niclosamide exposure within early embryogenesis. These graphs are super busy, but I want to draw your attention to what I have highlighted with the red circles. And what we saw here was increasing severity with earlier exposure initiation and a longer exposure duration, which is not super surprising. We did see significant recovery observed at these lower concentrations when the exposure was ended by 4 hours post-fertilization or when it was initiated at or after 4 hours post-fertilization. So what this suggested is that there's a potential sensitive window here between 3 and 4 hours post-fertilization. And interestingly, this exposure window coincides with some really important developmental events, including the maternal to zygotic transition, which is something I'm going to go into in a little bit more detail later. Right, so now we know what the phenotype is. Let's talk about mechanisms. So because niclosamide is thought to uncouple oxidative phosphorylation within invertebrates, our first question was, is it doing the same thing in these embryos? Is that what is causing this developmental delay that we're seeing? And so we strove to answer this question in 2 different ways. The first one was to look at oxygen consumption in exposed embryos. Oxygen consumption is a really common metric for looking at impacts to oxidative phosphorylation. And so to do this, we use closed vials with oxygen sensors on the bottom that allowed us to measure oxygen in that vial over a period of time. And so what you can see on this set of graphs over here, that top graph, we did some work to validate the correct sample size that we should be using for this assay. So we chose to use a sample size of 20 embryos. Then the niclosamide data is represented by this bottom bar graph here. What we saw is there was no significant alterations to oxygen consumption in these embryos following niclosamide exposure. And this suggested that maybe oxidative phosphorylation is not the main mechanism by which niclosamide is causing this developmental delay. So we also wanted to look at this in one other way. So impacts to oxidative phosphorylation result in inhibition of ATP production. So we wanted to see if we pre-treat these embryos with ATP, is that able to mitigate or recover this developmental delay that we're seeing? So we pre-treated with ATP, and it did not mitigate or block this niclosamide-induced epiboly delay. So this kind of added to that idea that this niclosamide-induced effect is probably not due to disruptions to oxidative phosphorylation. So if it's not due to the traditional mode of action, what's happening here? So we wanted to look at some potential other mechanisms. So we turned to some whole transcriptome mRNA sequencing, where we looked at the mRNA transcripts present in our niclosamide-treated embryos versus our control embryos. And those results here are demonstrated by this volcano plot. Essentially, what this is showing you is all of those mRNA transcripts. Those indicated in red are those significantly altered from our vehicle control. And as you can see, it had a widespread effect on the transcriptome. There's obviously a lot happening here. So to look at this a little bit closer, we pulled out the top differentially altered transcripts, both decreased and increased from that exposure. And although there's a lot of stuff going on in this table as well, I want to draw your attention to, there was a number of cytoskeleton transcripts that popped up here. And now we know, due to the literature, that impacts to the cytoskeleton can cause the type of phenotype that we're seeing. And so these results kind of suggested to us that maybe niclosamide is impacting the cytoskeleton, resulting in these developmental delays. And that was the hypothesis that we really were left with when we ended objective 2. So now I'll cover just a few conclusions from that. So we showed that niclosamide exposure during early zebrafish embryogenesis results in a concentration-dependent delay in this process of epiboly, with a potential sensitive window between 3 and 4 hours post-fertilization. It doesn't appear that niclosamide is altering oxygen consumption and suggests that this phenotype is due to something other than impacts to oxidative phosphorylation. And then our sequencing data showed that niclosamide results in widespread alterations to transcript levels, including many genes related to the cytoskeleton. So now for objective 3, we wanted to really dig in and investigate the mechanism of niclosamide toxicity a little bit more during early zebrafish embryogenesis. So when we were thinking about what other mechanisms could be at play here, we turned our attention to the literature. And epiboly delay is actually something that has been observed in embryos exposed to a wide range of compounds with a diverse range of different cellular and molecular mechanisms. And these mechanisms include mitochondrial impairment, alterations to the cytoskeletal microtubule and microfilament networks, impaired lipid utilization, as well as an inhibition of zygotic transcription. And so we wanted to address each of these hypotheses in turn. And I want to direct your attention to this flowchart I have on the screen here. This is kind of a way to kind of track where we are as we move through these various hypotheses. So the first thing we wanted to look at was the cytoskeleton. So before I jump into the data here, I want to just orient you a little bit on where the cytoskeleton is, where we can expect it during the stage of embryonic development. We have here, so we have 2 millimicrotubules that are present in the cell mass. We also have this lateral network of microtubules that is sort of stretching throughout the yolk sac. And these are responsible for kind of grabbing those cells and pulling them around the yolk sac during epiboly. We also have active microfilament networks that form a lattice around this cell mass up here, as well as a dense network of active microfilaments in the bottom of the yolk sac, and those act to maintain yolk sac integrity throughout development. So first we wanted to look at tubulin. Is niclosamide impacting our microtubules? And we addressed this question by fluorescently tagging tubulin within exposed and controlled embryos. And here's some representative images here on the screen. When we quantified this fluorescence throughout the various compartments of the embryo, we did not see any significant alterations to the distribution of tubulin within our treated embryos, suggesting that it doesn't appear that niclosamide is impacting these structures. We also wanted to look at this another way, so we used a mammalian in vitro tubulin polymerization assay. And essentially how this works is you have tubulin monomers in a test tube, you add some enzymes, fluorescent probes, and as those monomers polymerize to form an intact microtubule, you get an increase in fluorescence. So if niclosamide is impacting this process, we would expect to see a decrease in fluorescence. And that's what's represented on this bottom bar graph here. And you can see we did have a significant decrease in fluorescence. However, it's happening at concentrations that are way higher than what we're seeing in our zebrafish embryo. Therefore, it's really hard to try to extrapolate this to what we're seeing in the embryo. So collectively, from this data, we took away that disruptions to microtubules do not appear to be the mechanism that's driving this epithelial delay within zebrafish embryos. So moving on to the other part of the cytoskeleton, actin microfilament networks. We took a similar approach with actin, where we fluorescently take it in zebrafish embryos. Once again, there's some representative images for you. As opposed to our tubulin data, however, we did see significant decreases in actin within the zebrafish embryos. However, it wasn't everywhere in the zebrafish embryo. It was just in the cortical actin network. So you can kind of see it here by this white arrow. It's that bottom half of the yolk sac that's stated. And we saw significant decreases in that network there. However, we did not see significant decreases in staining in the cell mass. So it appears like it's not affecting the whole embryo. It's just affecting that one area. What we did notice, however, is that there was a significant increase in the size of cells in that cell mass. And this was really interesting because as the cells are dividing on top of the yolk sac during normal embryogenesis, those cell divisions are reductive, meaning that the cells dividing are maintaining the same space, the cells just get smaller as they divide. So an increase in cell size kind of indicates that maybe this embryo is experiencing a global developmental delay. Maybe this isn't just a specific impact on this process of epithelium. And so that was something that we thought that was kind of in the back of our head as we continued on with this project. So because niclosamide is really a human health concern, we also wanted to look at these impacts to actin in human embryonic stem cells. And that's what you're seeing here on this panel of graphs. These top two bar graphs are showing you cell survival or cytotoxicity. We did see significant impacts to cell survival. However, it occurred at concentrations about a 10-fold higher than those which we were seeing in the zebrafish embryos. And this suggested that for some reason, our zebrafish embryos were more sensitive to niclosamide within our particular experimental setup. We also take actin in these cells and counter-stain with DAPI. There's some representative images here on the bottom of the screen, and that data is represented by this lower bar graph. We did not see significant alterations to actin networks within these stem cells at non-cytotoxic concentrations. So this suggests that the effects we were seeing within the zebrafish to actin are probably due to other processes happening in the zebrafish rather than a targeted effect on actin networks. So moving on to the next hypothesis we wanted to address, which was, is niclosamide disrupting lipid utilization and resulting in epithelial effects that way? So to address this question, we used some non-targeted metabolomics to look at nonpolar lipids. And this data is represented by this heat map as well as this bar graph here on the screen. And essentially what we saw with all of this data is that niclosamide exposure did not significantly alter the relative abundance or distribution of nonpolar metabolites within these treated embryos. And this suggested that niclosamide-induced epithelial delay is probably not due to disruptions to lipid utilization. So moving on to our last hypothesis, which was inhibition of zygotic mRNA transcription. So before I jump into the data here, I'm just going to orient you a little bit. When zebrafish embryos are first fertilized and they're undergoing early embryogenesis, they're not actually transcribing their own genome. Instead, they are relying on a pool of maternally deposited mRNA that is translated within the embryo to produce the proteins necessary to drive early development and activate the zygotic genome. And that's kind of shown here to you in the schematic. So what we see is maternal transcripts present in the early stages. At around 3 hours post-fertilization, we get a decrease in maternal transcripts with an activation of the zygotic genome. Then you have a takeover by those zygotic transcripts. And this process is called the maternal to zygotic transition. So we wanted to see, is niclosamide impacting the activation of the zygotic genome in this process of the maternal to zygotic transition? So to answer this question, we again used mRNA sequencing to look at the relative levels of zygotic and maternal transcripts. There was a paper published back in 2013 that was able to parse out maternal and zygotic transcripts, so we were able to leverage that data set to divide up our own data. And so what we see with the controls, indicated in black here, is that as development is progressing, you have a decrease in these maternal transcripts with a simultaneous increase in zygotic transcripts, which is exactly what we would expect. However, interestingly, with our treated embryos, we did not see this. We saw a significant decrease in the amount of zygotic transcripts with an increase in maternal transcripts. And this was really exciting because it indicated that somehow niclosamide is impacting this maternal to zygotic transition. So we wanted to look at this a little bit closer. So we took these differentially altered transcripts and did a couple of different comparisons. So if you remember back to our active data and the significant increase in cell size we saw, we've been thinking that maybe this is really a global developmental delay. Maybe niclosamide is not actually targeting this specific process. And so to address that question, what we did is we compared the transcriptome of our 2 to 5 hour niclosamide exposed embryos to a time match control, so a 5-hour control. We also compared it to a stage match control, a 3-hour control, so an embryo that looks like a niclosamide treated embryo. And what you can see here from these volcano plots is that compared to our time match control, there was a widespread alteration to the transcriptome. However, when we compare it to that 3-hour stage match control, the magnitude of that effect goes way down. And this suggested that our 5-hour treated embryo is transcriptionally more similar to that 3-hour-old embryo, suggesting that, yeah, there's probably a global developmental delay occurring here. So to dig into that a little bit deeper, we took these differentially altered transcripts and took them into a gene ontology for biological processes, and that's what's represented by this bar graph here. Once again, there's kind of a lot going on, but I want to direct you to the bars that I highlighted. So for the for the time match controls, indicated in red, our top biological processes were related to transcription and transcription regulation. However, when you look at that stage match control, our top hit is for translation. And when you think about what's happening in these zebrafish embryos at this stage, this actually makes a lot of sense. Because if niclosamide is impacting the ability of these embryos to translate that maternal mRNA into proteins that it needs to complete development and initiate the transcription of the zygotic genome, we would expect to see translational hits compared to our stage match controls with transcription-related hits later. So we wanted to dig into that a little bit deeper. Right. So we had also done some polar metabolomics to look at significantly altered metabolites in these exposed embryos. That data is represented here in the box for you. So we had 31 altered metabolites following niclosamide exposure. The majority of those were amino acids, as you can see in the pie chart. When we took those altered metabolites through a pathway analysis, what we found was the highest altered pathway here was amino-acyl tRNA biosynthesis. And what this pathway does is it plays a crucial role in the translation of mRNA into proteins. So what we have here, a little schematic of what's happening during translation, is you have your maternal mRNA, you have tRNAs that are responsible for grabbing amino acids, bringing them to the ribosome, and incorporating them into that guiding polypeptide. And so this process is what we're seeing altered in our metabolite data. And so if niclosamide was altering this process somehow, that would explain our sequencing results and explain that developmental delay that we're seeing. So the last piece of data I'm going to show you today. When we got started thinking about this process, One thing that we thought about was, is niclosamide altering the synthesis of tRNA? Is that how this is happening? So to address that question, we quantified the relative contribution of tRNA. That's represented by this first paragraph here. We did not see any significant effects to the relative abundance of tRNA. We then went back to our polar metabolite data and looked at those significantly altered amino acids. And that's what's in these red bar charts here. And we did see significant increases in some cases to free amino acids. And this suggested that maybe amino acid utilization or amino acid recycling is being affected and altering this process. So this really represents kind of the state of the story as it is right now. So in that, I'm going to kind of recap on what we figured out from this objective. We found that exposure to niclosamide results in a global developmental delay. Overall, the data from all of these studies suggest that niclosamide is coming in, inhibiting the translation of maternal RNA into the proteins that are necessary to drive early development and initiate the transcription of the zygotic genome. And within this process, our data is suggesting that niclosamide might be interfering with the amino acyl tRNA biosynthesis pathway, or that it might be altering utilization of amino acids. All right, and with that, I'm going to take a step back and talk about some overall conclusions from this work. Overall, our findings from this dissertation, I think, really highlight the utility of the embryonic zebrafish as a physiological intact, non-mammalian model for the screening and prioritization of chemicals, as well as environmental samples. We demonstrated an unbiased way of progressing from a large library of chemicals in Objective 1 down to a specific compound hit with a previously unknown mechanism that has some real relevance to public health. And I think we also opened the door for future investigation into compounds that might be disrupting this process of the maternal to zygotic transition. This isn't really something that's talked about very often as far as a target for compounds. And I just want to end on highlighting that the maternal to zygotic transition is also not something that's conserved to zebrafish. It happens in a lot of different species, including humans, although the timing of that is different depending on the species in question. So with that, I want to just list some publications. I've been really fortunate to be involved in a lot of different studies during my time here at UCR. And I will jump into my acknowledgements. So first of all, Bowles Lab, my PI, Dr. David Bowles, has been the most supportive PI I ever could ask for. And the current and past Bowles Lab members, you guys are amazing. It's been a lab family, and I definitely couldn't have done it without you. I'd also like to thank the Schlenk Lab for being close collaborators over the years, for all of your help and support. Also, my past and present committee members, especially the three of you here today, as well as my collaborators, the Genomics Core, the Metabolomics Core, and Dr. Nicole Sparks and Dr. Zerneden for the help with the human embryonic stem cells. And then, of course, my funding that made all this possible. And I'm going to end by thanking my friends and family. I have an amazing supportive family that always supports my pursuits, even though they take me to weird places like California. I'd like to thank my in-laws and especially thank my husband Alex for always being supportive, always being there to hear me vent about all of the science. You know, it doesn't always go right. I'd like to thank my friends. I've been super lucky at UCR to have an amazing group of friends that have really become my family away from home. And thank you all for your support. And I couldn't end a presentation without putting Sushi Cat on here, so there she is. And I will end today with any questions and comments and a few pictures of where I'm off to my new adventure. I accepted a postdoc with the US EPA in Duluth, Minnesota. Adventure. Thank you.
Okay, so Sarah can take some questions from the audience, and then we'll have a close session. You always have questions.
30, slide 31. Can we look at that?
Guess you were taking that.
My brain. Okay. All right. I'm just new to these graphs. You said that there's no change, but what I see is, especially in that bar graph, there's almost like an order of magnitude change there, difference in one of your one of your
Yeah.
concentrations.
No, so there definitely was a trend here. It just, I think the variability here was so high that we didn't get any significant hits.
Okay, I see, the variability,
Mhm.
okay. Even with
Yeah.
that.
And this is the relative abundance of non-polar lipids. So essentially just summing up everything and
You've emphasized, I mean, it's actually an impressive piece of work you've done. Because you emphasize the effect of this compound on the translation of maternal mRNA. Does the same thing not happen with the RNA of the child, the developing embryo?
yeah. No, yeah, that's a really good question. And it certainly can. Now let me go to this slide real fast. Oops. Yeah, no, so it's certainly possible that niclosamide could be impacting translation as a whole, right? Not just targeting those mRNA transcripts. The reason that we're focusing on that is because at these stages, we're really not seeing levels of the zygotic transcripts emerging here, right? So at this stage, development is really being driven by those maternal transcripts, which is why we think it might be targeting those.
It might be helpful maybe to go to the recovery
Sure.
sensitive window slide, because that would suggest that it is specific to the maternal side.
Yeah, so Dave's exactly right. So the fact that we see a potential sensitive window between 3 and 4 suggests that this process of maternal to zygotic transition is what's potentially being targeted here. So we see significant recovery when exposures were initiated after 4 hours, which is when you see that zygotic transcription occurring. So I guess if niclosamide was targeting the translation of everything as well, we would probably not see that recovery.
Do you think there's a difference between the maternal and the zygotic?
I don't think there's a difference. I think it's a difference of abundance. I think because those early stages are primarily driven by maternal mRNA, like that's what's there. I think that that's what's there for niclosamide to be targeting. One of the things I wish we could have done more with this was look at later stages. And I think that would have been really interesting to see what was happening when those maternal transcripts were degraded and the embryo was relying solely on zygotic mRNA.
Does the niclosamide that's used for the tapeworm treatment in animals, that have a high risk of getting into wastewater and then making its way to fish populations and breeding grounds?
Yeah, so it is excreted. However, it doesn't stick around very fast. So it undergoes photolysis super fast in bodies of water. So I don't really expect it to be much of an environmental concern for that reason. Yeah, it was actually used as a pesticide on and off.
Any other questions? Okay, thank you, Sarah.
Oh, actually, I like that.